Welcome back, class. My name is Silvana O'Coney and today's lecture is lecture 6, the fungi. In this lecture we'll introduce the group of eukaryotes called fungi. We'll look briefly at their distribution, importance, structure, nutrition and metabolism, reproduction, and the 8 major fungal subdivisions. Now we're going to look at the major fungal subdivisions in the kingdom fungi. Here are the 8 main subclasses that we'll be looking at, as well as some of the characters involved in their classification. First we have the chytridiomycetes, followed by zygomycota, ascomycota, basidiomycota, uridiniomycetes, eustolaginomycetes, glomeromycota, and microsporidia. Here we can see the first 4 subclasses, the major characteristics that are used to classify fungi within each class, and also some representative examples on the right hand side. So note that Rhizopus, our black bread mould that we just looked at, is found in the second subclass, Zygomycota. In the third subclass, Ascomycota, we have the yeast Saccharomyces and also the famous Penicillium mould. Many of our common mushrooms, including Agaricus, are found in the 4th subclass here, Basidiomycota. And if you have a bit of a read through here, you'll note that the type of spores produced by each fungus, whether asexual, sexual spores, and the type of spores, figures quite heavily in terms of classification. Here are the other 4 subclasses. Note that many of the plant pathogens called rusts are found in the uridiniomycetes, with intracellular fungal parasites of animals found in the last subclass listed here, the microsporidia. The simplest of the fungi are the chytridiomycetes, or simply chytrids. They're actually quite unique among the fungi because they produce a zoospore with a single posterior whiplash flagellum. And this is considered a primitive feature, that is the presence of a flagellum that was lost in the more evolved fungi. Free-living members of this group are saprophytes, that is, they live on plant or animal matter in fresh water, mud or soil. Parasitic forms can infect aquatic plants and animals, including insects, and a few members of this group are found in the rumen of herbivores. The chytrids display a variety of life cycles, including both asexual and sexual reproduction. Members of this group are microscopic in size, and they may consist of either a single cell, a small multinucleate mass, or even a true mycelium with hyphae. Sexual reproduction results in the release of sporangiospores from sporangia, and many members of this group are capable of degrading cellulose and even keratin, which enables the important degradation of crustacean exoskeletons. Our next group of fungi are the zygomycetes. Most of the zygomycetes are saprophytes, that is, they live on decaying plant and animal matter in the soil. A few are parasites of plants, insects and other animals, including humans. The hyphae of the zygomycetes are cenocytic hyphae, which contain numerous haploid nuclei. Asexual spores develop in sporangia, and sexual reproduction produces those tough, thick-walled zygotes called zygospores that we saw for Rhizopus stolonifer. So we've looked at reproduction by Rhizopus stolonifer, the bread mould, but this genus Rhizopus is also important because it's involved in the rice disease known as seedling blight, which you can see in the photo here. And if you consider that rice feeds more people on Earth than any other crop, the implications of this disease are quite obvious. It was thought that the Rhizopus fungus was secreting the toxin that kills these rice seedlings. So scientists started researching the toxin and the genes that encode the toxin, but much to everyone's surprise, they actually found that a bacterium called Burkholderia was found growing within the fungus, and it was the bacteria that was producing the toxin, not the fungus itself. So with another interesting twist to this story, that same toxin that kills the rice seedlings has been shown to stop cell division in some human cancers, and it's now being investigated as an anti-tumour agent. The zygomycetes also contribute to human welfare. So, for example, one species of Rhizopus is used in Indonesia to produce a food called tempeh from boiled soybeans. Another zygomycete is used with soybeans in Asia to make a curd called sufu. And still other members of this group are employed in the commercial preparation of some anaesthetics, birth control agents, alcohols, meat tenderizers, and even the yellow colouring used in margarine. The subclass Ascomycota contains fungi called ascomycetes, and they're commonly referred to as the sac fungi. This group of fungi are ecologically quite important in freshwater, marine and even terrestrial habitats because they degrade many chemically stable organic compounds including lignin, cellulose and collagen. Many species will be quite familiar to you and also economically important. So for example, most of the red, brown and blue-green moulds that cause food spoilage are ascomycetes. The powdery mildews, such as this powdery mildew growing on a pea plant, pictured here, these are normally fungi that belong to this group. Even the fungi that cause chestnut blight and Dutch elm disease are ascomycetes. Many of the yeasts, as well as edible morels and truffles, are also ascomycetes. The pink bread mould pictured here, Neurospora crassa, is also an ascomycete, and this is an important example of a research tool used in genetics and biochemistry. Many of the ascomycetes are also parasites on higher plants. For example, the ascomycete called Claviceps purpurea parasitizes rye and other grasses, and it causes the plant disease called ergot. Ergotism, the toxic condition in humans and animals who eat grain infected with this fungus, is often accompanied by gangrene, psychotic delusions, nervous spasms, abortion and convulsions. During the Middle Ages, ergotism, also then known as Saint Anthony's fire, killed thousands of people. For example, over 40,000 deaths from ergot poisoning were recorded in France in the year 943. And it's also been suggested that the widespread accusations of witchcraft in Salem and other New England communities in the late 1690s may have resulted from outbreaks of ergotism. The activities are due to an ingredient called lysergic acid diethylamide, or LSD. Here we have some more examples of ascomycetes. In A, we have the common morel, Morchella esculenta, which is one of the choicest edible fungi. In B, we have some scarlet cups, and in C, the prized black truffle, Tuber brumali. And the black truffle is highly prized for its flavour by gourmet cooks. And truffles, of course, are mycorrhizal associations with the roots of oak trees. Many of the ascomycetes are yeast. The term yeast refers to unicellular fungi that reproduce asexually by either budding or binary fission. Here we have the life cycle of the yeast called Saccharomyces cerevisiae, and this life cycle is particularly well understood. Saccharomyces cerevisiae alternates between haploid and diploid states. As long as nutrients remain plentiful, haploid and diploid cells undergo mitosis and budding to produce haploid and diploid daughter cells respectively. Let's look at these. Have a look to the right of this image where it says vegetative life cycle haploid. So these yellow cells that you can see here are haploid alpha cells, and they're reproducing asexually by budding. To the left of that figure, we can see orange yeast cells called A cells. These are also haploid cells, and they're also reproducing asexually by budding. To the bottom of the figure, we have some reddish brown cells, yeast cells, and they are also reproducing asexually by budding, but these cells called A alpha are diploid cells. Now, if you refer to the photo to the left of screen, we can see budding occurring. Each daughter cell leaves a scar on the mother cell as it separates, and you can see 3 scars on the mother cell. They're called bud scars. And each new daughter cell will only bud from an unscarred region of the cell wall. When a mother cell has no more unscarred cell wall remaining, then it can no longer reproduce and it will die. Okay, let's go back to the figure on the right hand side. So we've got the yellow alpha cells reproducing asexually, the orange A cells reproducing asexually, and also the reddish brown diploid cells reproducing asexually. Now, what happens when nutrients are limited? Then the diploid reddish brown A alpha cells to the bottom of this figure will undergo meiosis. So now we can follow the black arrows towards the top of the figure, where one of these diploid cells has undergone meiosis to produce 4 haploid cells that remain bound within a common cell wall called the ascus. And the ascomycetes are named for this reproductive structure, the sac-like ascus. Now, you can see that this particular ascus contains 4 haploid ascospores, 2 yellow alpha cells, and 2 orange A cells. These are released eventually from the ascus. They germinate and continue the vegetative life cycle of haploid cells. Now, on the addition of nutrients, if 2 of these haploid cells of opposite mating types, that is an A cell or an alpha cell, If they come into contact, they will fuse together to create a diploid cell. And typically only cells of opposite mating types, as in A or alpha cells, can actually fuse together. And this is a process that's very tightly regulated by the action of pheromones. Next we have the Basidiomycota, which includes the Basidiomycetes, commonly known as club fungi. Examples include jelly fungi, rusts, shelf fungi, stinkhorns, puffballs, toadstools, mushrooms, and bird's nest fungi. This group of fungi are named for their characteristic structure or cell called the basidium, which is involved in sexual reproduction. The Basidiomycetes affect humans in many different ways. Most of them are saprophytes that decompose plant debris, especially cellulose and lignin. So, for example, the common fungus called Polyporus squamosus forms these large shelf-like structures that project from the lower portion of dead trees, which they help to decompose. Many mushrooms are used as food throughout the world. The cultivation of the mushroom Agaricus campestris, pictured here on the right, is a multi-million-dollar business. And of course, not all mushrooms are edible. As suggested by its name, ingestion of the mushroom Russula emetica induces vomiting. Many mushrooms also produce specific alkaloids that act as either poisons or hallucinogens. One example is the death angel mushroom, Amanita phalloides. The toxins produced from this mushroom attacks cells lining the stomach and the small intestine, and they're responsible for the severe gastrointestinal symptoms associated with mushroom poisoning. Some of the fungi belonging to this group are pathogens of humans, other animals, and even plants. One example is Cryptococcus neoformans, which is an important human and animal pathogen. It produces the disease called Cryptococcus, which is a systemic infection primarily involving the lungs and the central nervous system. Here we have the life cycle of a typical soil basidiomycete, which actually starts with a basidiospore germinating in the soil to produce a mycelium. And we can see two germinated basidiospores, a plus basidiospore in blue to the bottom left of screen, and a minus basidiospore in pink right next to it. Now these quickly grow and spread through the soil, and when they meet each other and they're opposite mating types, which these two are, then they fuse together to form a new dikaryotic mycelium. Eventually this is stimulated to produce basidiocarps. A solid mass of hyphae then forms a button, which you can see pictured here and labelled, and this button pushes through the soil, elongates and develops a cap, or in other words, the mushroom that we're all familiar with. Now the cap of the mushroom contains many plate-like gills, each of which is coated with basidia. And you can see an enlarged portion of the gill covered with the basidia at the top right of screen. The two nuclei in the tip of each basidium fuse together to form a diploid zygote nucleus, and this immediately undergoes meiosis to form 4 haploid nuclei, also indicated here. These nuclei push their way into the developing basidiospores, which are then released at maturity, and the life cycle commences once again. here we have the two subclasses, Uridiniomyces and Eustalarginomycetes. Both include very important plant pathogens which cause rusts and smuts. In addition, some of the Uridiniomycetes include human pathogens. Now, these fungi are very virulent plant pathogens that cause a huge amount of damage to cereal crops. Millions of dollars worth of crops are actually destroyed annually. The Eustalarginomycete, called Eustalargiomatus, is a common corn pathogen that's become a model organism for plant smuts. It's dimorphic, and the yeast-like saprophytic form can be easily grown in the laboratory. In this photo, we can see the pathogen, Eustalargio, which causes tumour formation in corn. So note the enlarged tumours, which when mature, they'll release masses of dark teleospores in place of the normal corn kernels. This is eaten as a delicacy in Mexico and called huitlacoche, also referred to as the Mexican truffle. The fungus is actually worth more than the corn itself, and it contains far more protein. It's exported from Mexico to the USA. The glomeromycota are of critical ecological importance because most of them are mycorrhizal symbionts of vascular plants. Mycorrhizal fungi, as you know, form important associations with the roots of almost all herbaceous plants and tropical trees. And of course, this is considered a mutualistic relationship because both the host plant and the fungus benefits. The fungus helps protect its host from stress and also delivers soil nutrients to the plant, which in turn provides carbohydrate to the fungus. Only asexual reproduction is known to occur in the glomeromycota. Spores are produced and they germinate when in contact with the roots of a suitable host plant. Our last group of fungi is the Microsporidia. And out of all the fungi, this group has the most confused taxonomic history. They were widely considered protists, and they're still referred to as protists by some people. Unlike other fungi, they lack mitochondria, peroxisomes, and centrioles. But importantly, they are obligate intracellular parasites that infect insects, fish, and humans. To the right of screen, you can see the little red arrowheads, which are indicating masses of Microsporidia in infected muscle tissue in Pacific white shrimp. And in the photo to the bottom of screen, the white arrow is indicating masses of large cysts of Microsporidium spores in monkfish. Microsporidia morphology is also unique among the eukaryotes. Small spores of 1 to 40 microns are viable outside the host. Depending on the species, the spores may be spherical, rod-shaped, egg-shaped, or crescent-shaped. Spore germination is triggered by a signal from the host cell, and it results in the expulsion of a tightly packed organelle called the polar tube or filament, which you can see in this figure at the top right of screen. The polar tube is ejected with enough force to pierce the host cell membrane, which permits the parasite's entry. And then once inside the host cell, the Microsporidium undergoes a developmental cycle that differs depending on the species. However, in all cases, more spores are produced and eventually take over the host cell. So although you can imagine this would have fairly catastrophic consequences for humans and animals, the use of microsporidia that infect destructive and pathogen-bearing insects is actually under development as a biocontrol measure.